recording, and so we are recording right now. Let me just do this. So I'm going to get right at it today. If you need to ask me something while I'm talking, that's fine. I don't mind if you do it audibly. You can also just text me a message and I'll address it. It pops up on my screen and I can address that. So first thing I'm going to do is get to the nervous system. This whole last section of the semester, a lot of it, most of it's going to be dedicated to the nervous system because there's so much material in the book. It's very complicated. We can't possibly cover all of it. It's just not possible. It would take us, most of us, some of a regular 16-week semester to cover the entire thing. So I have highlighted on the outline exactly, or that I'm going to publish today, as well as I put an announcement out about the last 4 quizzes for the semester. That's already posted on our Canvas page, and then later today I'm going to post a detailed day-by-day outline. And I chose the sections, there's multiple chapters about the nervous system. I chose the sections that I think will best help you with your future studies, with what you do after A&P 1 at Camden County College. So without further ado, I'm going to start this right away. So we're just going to do the basics of the nervous system for the next couple Zoom lectures. And the first bit that we start off with is just, there are 3 functional elements to the nervous system. Okay? And what those 3 functional elements are, basically there's incoming information, everything comes into the central nervous system, the brain and spinal cord. So we've got an incoming pathway, then we've got a processing plant, which is our brain and spinal cord, and then there's an outgoing response pathway. That's what the 3 functional elements really are in layman's terms. So I'm just going to write these out as your book does. The incoming pathway is just called sensory input. Okay. All right, I'm just trying to, Patrice, if you could please mute your microphone. We can hear what's going on. I appreciate it. Thank you. So the sensory input this is the incoming pathway of information from sensory receptors. This is typically the first step of the pathway of the nervous system. Now, sensory receptors can sense the external environment as well as the internal environment, but there's always some sort of incoming pathway to the central nervous system. Then, actually, the central nervous system is what we call the integration center. So I'm just going to write integration because your book uses that. That's a T, by the way. Uses the word integration a lot. So this is just the processing of number 1, sensory information. Okay, and then the brain and spinal cord, that integration center, processes the information, and then it sends out some sort of outgoing command, which we call motor output. The motor output is the response to this thing, the sensory input. So we always start with some sort of sensation, sensory input. It goes to the central nervous system where that information is integrated, processed, and then some sort of response to number 1 is sent out via a motor output. So I'm just going to put, this is the response. This command is to the response to the receptors from number one, and it is sent to what we call effector organs. Classic example, ready? Here we go. You walk outside in February and it's really cold, but you're only wearing a t-shirt and shorts. Your sensory receptors, mostly in your skin, sense the change, that cold. The environment is super cold. Those receptors send a signal to the integration center, which is the central nervous system, the brain and spinal cord. Your brain and spinal cord processes the information and says, that's where it makes sense of it, and says, it's really cold. If we don't do something, the internal body temperature is going to drop, and that's not good. So then the integration processing center sends out a motor output or a response to the receptors sensing cold, and the motor output is sent to effector organs. In this example, the effector organs would be muscles all over the body that start to shiver. When we shiver, when we're cold, that's actually muscular contractions, and that's a response in order to raise our internal body temperature. So that's an example of how the nervous system works. I've said this already. I'm just going to write central nervous system, or CNS. When we use this term CNS, you know now that this is just going to refer to the brain and spinal cord. This is the processing plant of all incoming information. It's also what sends out a response via the motor output. So words like integration, processing, control center, all of those things have to do with the central nervous system, the CNS. So it interprets incoming sensory information and then dictates some sort of motor output. These are anatomical terms. Central nervous system just refers to brain and spinal cord. I just added the functional elements of that. The peripheral nervous system is also an anatomical term that we abbreviate PNS, and that refers to basically everything outside of the CNS, everything that's part of the nervous system that's not part of the brain and spinal cord. It's mostly neurons, it's mostly nerves emanating from the spinal cord that radiate out into our appendages, hence the term peripheral. So this is actually acts as the links to the central nervous system. So the peripheral nervous system is basically the sensory input and the motor output. That's what the peripheral nervous system uses to accomplish the task of number 1 and number 3. There are two major subdivisions of the peripheral nervous system, and they are functional subdivisions. And if you look up at number 1, 2, and 3, that's what I'm referring to, numbers 1 and 3. The first one we refer to as the sensory, and we use this word all the time in conjunction with it, afferent, with an A, the sensory afferent division. These two words will always go with each other, sensory and afferent. Okay, this division carries sensory input toward the central nervous system. That's by definition. By just writing towards CNS. I have to layer over this board, and I'm just gonna, there's two arms of this sensory afferent. So I'm gonna erase this, not because it's not important, but just so I can squeeze in. There's two arms to this sensory afferent division. Just part of this monitors the inside world and part of it monitors the outside world. So there's a somatic, sorry, I had my eraser, I meant to have my black pen. So I'm going to put letter A. There is a somatic sensory afferent nervous system, or fibers, and these are from things like the skin. They monitor the external environment. So things like the skin, skeletal muscles, and even joints. And then there is an arm or part of the sensory afferent division called the visceral sensory nerves or fibers. That means internal organs, visceral. The viscera really means the organs within our abdominal cavity, but this is all encompassing. So this visceral would be things like, I'm just going to put all visceral organs. Okay. So before I leave this page, before I layer over this, I just want to review real quickly. These are functional divisions of the entire nervous system. This just defines anatomically what the central nervous system is. This, we haven't really defined anatomically. It's just all of our nerves and the peripheral nervous system outside of the CNS. Now we're talking about functional subdivisions of the peripheral nervous system. So before I leave this page, I wanted to reiterate that we've done number one, a sensory afferent division or afferent. So I need to layer over this and go to number two. There is a motor efferent with an E. Division. Within that, we're still talking about the peripheral nervous system. These two words also go together, motor and efferent, just like sensory and afferent on the previous page will always be linked with each other. So this carries motor impulses away from the CNS. This is what's going to carry all of the commands that the central nervous system issues in response to the sensory input. And there's what we call the somatic arm of the motor efferent division. Sometimes some books will just simply call it the somatic nervous system. But this somatic part of the efferent division is to voluntary skeletal muscles. Okay? That's important to remember. And then there is something called the autonomic nervous system. I'm sure you've heard of that. This is to involuntary muscle, aka cardiac, and smooth muscle, and also glands. This one is voluntary. This one is involuntary action that we don't have conscious control over. So there's kind of an anatomical and functional breakdown of how this nervous system work. Just to make things even worse, more complicated, this one has a parasympathetic division. This is why this is so complicated. And there's so many chapters within your textbook. And a sympathetic division. Talk about those much later in this nervous system unit. These are both subdivisions of this subdivision, which is another subdivision. So we're just layering on more complication. This controls our everyday body functions. Like you just ate breakfast, your digestive system is digesting, your breathing rate, your blood pressure, all that business. That's why it's called the rest and digest part of the nervous system. This one is referred to as the fight or flight. This kicks in when we are stressed out, and it overrides this one, which does the basic maintenance of body functions. All right, now I need to move on. There's so much to cover today that I know I'm going quite quickly. If you review this video or you find that you're not really understanding what you are reading in your textbook, just ask me a question anytime. I don't mind explaining it. I just don't have time to go through every word in this textbook. There's just too much. We don't have time. So I get it that I'm moving a little bit quickly. That's because I have to cover so much ground. I'm writing nervous tissue. And this is sort of the next topic, I believe, in your textbook. I'm trying to go in the exact same order. We mentioned nervous tissue when we studied tissues way back in the first unit. And we did mention that there are two different major classes of cell types. So I'm going to reiterate that right now, and we're going to talk much more in detail about them because we just glossed over that in the tissue section. Neurons and neuroglia are the two types of cells. If you remember, these are the money cells that transmit electrical impulses. These are helper cells. And there's a whole bunch of those. Can you type that, flash that message up again, and I'll answer the question, no problem. It disappeared before I could read the whole thing. Thank you. It was something about efferent and afferent. I'd be happy to explain it. So these cells I'm going to talk about briefly. Okay, so I just had a question that everyone needs to hear the answer to this. Both afferent and efferent have somatic divisions that involve skeletal muscles. Sort of yes. So the short answer is yes to that question. The long answer is yes, they both involve skeletal muscles, but somewhat differently. We have sensory nerves, part of the sensory afferent. Sometimes I say afferent, just so you remember that that's an A and an E. The pronunciation can confuse us as well. But they both involve skeletal muscles in the somatic division, but they're different units of the nervous system. So our skeletal muscles, for example, have sensory receptors in them that are part of the somatic sensory fiber system that send impulses toward the central nervous system. And there's also command fibers, directive voluntary command fibers that go back toward skeletal muscles to enable them to contract. So we have sensory receptors embedded in skeletal muscle that we call somatic sensory fibers. And then we also have somatic fibers running from the brain and spinal cord to skeletal muscles that control them to contract and behave. Before we, you know what, this would be worth it for in my opinion. I'm going to come back to this after 2 minutes. This will help everybody with the entire nervous system. So down here, I'm going to draw a map of the nervous system in about 2 minutes. We have, I'm going to write sensory, afferent, receptors. And we have these both monitoring the external world as well as the internal world. So I'm going to write somatic and visceral. Okay. They send impulses toward the central nerve. They're part of the peripheral nervous system, this path that I'm drawing right here. So this is all nerve fibers going toward the central nervous system, which is the brain and spinal cord. And this is where we process information, where there's integration. By the word processing, we mean, let's just say you touch your finger to a hot stove. Here, there are sensory receptors in the skin, they would be somatic, in the skin of your finger that sends an impulse to your brain. Your brain processes that sensory information and says, heat. We better do something about that or there's going to be tissue damage. And so then your brain and spinal cord sends out a command this direction. And the reason I'm going back through PNS is now this is still part of the peripheral nervous system. We call this the motor efferent or efferent division that sends an impulse away in response to this initial, you touch your finger on a hot stove. And this pathway now from your brain sends an impulse that we call through the motor efferent division to the somatic arm of that efferent division, which is skeletal muscles, voluntary skeletal muscles, and it tells them to pull your finger away from that hot stove. So all of the muscles in your arm and hand contract, and that all happens in microseconds because it's so fast. You think it's involuntary, but technically it's not. This particular example that I'm giving you. But so that's a classic example of finger on a hot stove, receptor send an impulse to the brain. The brain says, better get your, that's heat, tissue damage. Unless we do something, it sends an impulse to all the muscles in your arm to pull your hand away from that stove. But the same example can be given, let's just say you ate some food. There are sensory receptors in your stomach, so I'm underlining visceral sensory receptors, that sense food in the stomach, both by stretch receptors in the wall of the stomach, as well as chemical receptors, chemoreceptors that sense a change in pH in your stomach. And they, guess what? They send a signal, in this case, not all the way to the top of your brain, just to the brain stem, but still part of the central nervous system. And your central nervous system, your brain stem says, there's food in the stomach. And it sends out then a command via the motor efferent division, but this time it's not going to voluntary skeletal muscles. It's going to the autonomic division, involuntary. And in this case, my food example, to the parasympathetic division of the autonomic nervous system. This is known as rest and digest, and it tells your stomach to start secreting things and churning. So I'm just going to write rest and digest. And by churning and secreting things, it starts to digest your food. The last example I'm going to give is this. Now let's say this is a sort of a cheesy but very typical example that textbooks give. You're walking down a dark alleyway at 2 a.m. and something heightens your senses that you're fearful for your life. And that those sensory receptors start here, they go to the brain, your central nervous system clicks on and says, it stresses you out. This is a dangerous situation. And it sends out your brain and spinal cord, central nervous system then sends this same pathway, but down this arm or subdivision of the autonomic nervous system to your sympathetic. And the sympathetic always overrides what's going on. So you're walking down the street and this is what is almost always turned on in all of us, the parasympathetic rest and digest. But in specific, stressful, oftentimes life-saving situations, the sympathetic overrides all of this stuff and then It affects the same organs within us, but it does the opposite. It does things in order for us to fight or flight for our lives. So it turns on all of these life-saving operations. It restricts blood flow to certain areas and dilates it and shunts it to other areas. It dilates pupils, increases breathing rate, increases blood pressure, all sorts of things that we need to do to either fight for our lives or run for our lives. So that's basically right there. This map is how the nervous system works. And if you understand this at the get-go, all of the detail that we can layer on over this will make more sense, because it's always going to act like this. It's always going to do this, or it's going to do this, the nervous system. Or finally, it's going to do this. All right, now I'm going to go back to nervous tissue and quickly. So this is a continuation of nervous tissue. I'm going to talk about neurons. We know they're the money cells. They initiate and transmit electrical impulses. But there are some general characteristics of these cells that we need to talk about first. You know what, I think your book does neuroglia first. So I want to be consistent so you don't get confused. Let's do these helper cells first. There's 6 types, and we classify them. There's 4 helper cells in the central nervous system and 2 helper types of cells in the peripheral nervous system. So let's just quickly go through those. So I'm talking about neuroglia. I'm just going to list them 1, 2, 3, 4, 5, 6. 1 through 4 are going to be these. 5 and 6 are going to be these. So the first type, and I'll try to go in the order of your book, are astrocytes. These just get their name because they look like stars. They have little spikes coming off them. When they were first discovered, scientists thought they looked like space stars in the heavens, in the sky. So that's how they got their name. That's not critical to their function. So they are star-shaped, but I'm not really going to ask you that. I'm going to expect you to know this. They're the most abundant neuroglial cell in the central nervous system. That's first. They function in physical support. So they help construct physically and help make up the framework of synapses. So they support and frame, I'm just going to write framework for those junction areas called synapses. They also recapture or recycle potassium ions. So I'm writing a K+. That will be more relevant to you after we go through how neurons actually work themselves. So this is during the act of depolarization. They can recapture and reuse potassium ions. They also recycle neurotransmitter, which is the chemical used to send, that's why it's called transmitter. It's the chemical used to transmit a nervous impulse. They help process brain information. And finally, the last thing I'm going to say about these is they can signal. They do have the ability, they don't send electrical impulses, but they can signal or communicate with each other through calcium pulses, calcium ion. They send calcium ion pulses and can communicate with each other. All right. Second type of neuroglial cell, or sometimes they're just called glial cells. That's an R, M-I-C-R-O, microglial cells or microglia. These have, just to give you a comparison in physical look, they also have thorny processes to them, but they're not actual star-shaped like this. So astrocytes kind of look like this. Microglia tend to just have small little thorny processes on them. And they're smaller, hence their name, microglial cells. These are phagocytic cells. That means they act like immune cells. They have the ability to engulf and literally devour foreign debris, old broken down organelles and tissue. So they're phagocytic cells. They also phagocytize, I'm just going to put old, worn out nervous tissue or cells. They can also phagocytize foreign microorganisms. That's why it is said these cells act like immune cells. We don't want any foreign cells, foreign microorganisms to enter our central nervous system. That's from the get-go, but you know nothing's perfect, and occasionally bacteria or viruses do get into our central nervous system, and these cells, in particular microglia, help to identify those foreign cells and phagocytize them. I'm just going to layer over this one, go to number 3. Still talking about neuroglial cells in the central nervous system. Ependymal cells. Some people say ependymal. I learned to say it as ependymal cells. These line the central cavities of the CNS. Of the brain and the spinal cord. And what they do is they have processes on, so the cells kind of look like this. They have these processes on them. They're like cilia or flagella, and they help circulate fluid, cerebrospinal fluid. And finally, number 4 in the central nervous system are oligodendrocytes. You've heard all of this before at some point. Most of this won't be foreign to you, but if you haven't heard this language before, these actually produce, they're responsible for producing, sorry about that spelling, they form the myelin around neurons in the central nervous system. I'm just going to put around fibers in central nervous system. Okay, these all 1 through 4, remember, are central nervous system cells, neuroglial cells. Now I'm going to put a little break here and just write number 5. 5 and 6 are going to be in the peripheral nervous system. Number 5, satellite cells. Now we're out in the periphery, away from the brain and spinal cord. These sort of behave like astrocytes in the central nervous system. So they surround neuron cell bodies, which are called soma, by the way, and I'll reiterate that when I go through the general anatomy of a neuron. But they surround neuron cell bodies in the peripheral nervous system, and they function, they have a structural function, just like astrocytes. They also support and help form synapses. In the PNS. And finally, the last one are Schwann cells, which mimic or are very similar to, These are also found in the peripheral nervous system, but they behave just like these. They form a myelin sheath around fibers in the peripheral nervous system. These are lipid-based or fat-based cells in nature. They help with transmission and insulation. I'll talk about myelin as a product later. So those are the 6 glial cells. Now I'm going to move on to those. Those are helper cells. We find 1 through 4 in the central nervous system, 5 and 6 in the peripheral nervous system. Now I'm going to talk, most of what we talk about after this moment is going to be about this type of cell for the whole rest of the chapter. Neurons. These are the cells that initiate and propagate electrical impulses. But a few general characteristics about these, as opposed to, say, other cells in the human body, what makes them unique. First, they have very long lifespans. And you'll see why that's important, because number 2 characteristic is they are amitotic. That means that the cells cannot divide. And once a neuron dies, pretty much it's gone for good. That's why it's a good thing that they live for 80 to 90 years if something else doesn't happen to us. Is because they do not have the ability for cellular division. And so then basically the neurons that we form early in life are the neurons that we have for the rest of our life. And finally, number 3, they have a super high metabolism. I'll mention this again. They have a high metabolism because they're constantly producing that chemical neurotransmitter. So they need a constant supply of oxygen and an energy source like glucose. So the whole reason really that we breathe every second, almost 24 hours a day of our entire lives, is because one of the big reasons is because we have such large brains compared to other animals, and it takes up this structure, this brain takes up roughly uses about 30% of all cellular metabolism. So all energy in our body, 30% of it goes toward this organ, the central nervous system. And these cells have such high metabolism that the demand for oxygen and a constant energy source is huge, which is another reason why it's theorized why we as humans carry so much adipose tissue, so much fat, not only around our organs, but under our skin, is because that's a constant energy source. Not necessarily glucose, but our bodies go to breaking down lipid fat cells when we don't have an immediate source of sugar. So this thing uses so much energy that we need reserves as well all the time, and we need to constantly breathe all the time. Now I'm going to talk about the anatomy of a typical neuron. So this is just neuron anatomy. It's all in your book, but I'm going to try to simplify it for you. You'll see me draw this neuron a lot, multiple times throughout this unit. I'm going to draw this nerve cell. This is a typical motor neuron in the human body. And I'm going to label all these parts and briefly tell you their functions. This area I'm going to outline in dark black is referred to as the cell body of the cell, or soma. And that circular structure is just the nucleus inside of the soma. So there we've got a cell body called the soma and a nucleus within that cell body. Now we're going to label other parts of this, and let me label the nucleus just so there's no confusion. So there's genetic information in these cells. Unfortunately, though, they can't divide. They don't go through mitosis. I just need to clean up my drawing for a minute. This cell is constantly metabolically active, and it's producing proteins and chemicals at a constant rate. So in the cell body, we find typical things like mitochondria, ribosomes, endoplasmic reticulum that's associated with the nucleus. All those typical organelles are found in here. And usually what they're doing, spend all of their time doing, is cranking out the raw material for neurotransmitter, for chemicals. And that raw material is sent all the way down to the end here, and then it's assembled. And so in these ends here, which are called axon terminals, that neurotransmitter, I'm going to draw a really big axon terminal. Looks like this. The neurotransmitter is assembled, packaged, and stored in these bubbles called vesicles that I drew when we did the muscular system. So that's what's going on inside of an axon terminal. Now, other pieces to this puzzle or this anatomy, when I say the word, or you read the word process, I know that word has dual meaning. In this case, when we're talking about the central nervous system processing, that means doing something with information. In this anatomical context, the word process just means branch. And these thin processes that are attached to the cell body are called dendrites. So I'm just going to write up here, process in this case means branch or extension. So a dendrite is a process that's attached to the cell body. All of these little sticks that I've drawn are dendrites, and they're attached all the way around a typical neuron cell body. What they do is they receive information and send it toward the cell body. So I'm just going to write the word receive under dendrite. And then this long, big process here that I'm highlighting or making bold is not a dendrite, it's an axon. And that axon does the opposite functionally. It sends electrical information away from the cell body. Now, most neurons have many dendrites. Some of them don't, some of them only have one. But most neurons have one or more dendrites that receive information. All neurons only have one axon. So there can only be one axon on any given nerve cell, no matter what type it is. But there can be one or many, many dendrites that receive information. So just to reiterate, in here we've got mitochondria, ribosomes, rough endoplasmic reticulum. This whole cell body, the soma, with its dendrites, acts as a receiving area. So I'm going to write that. So the receiving area of the cell is here. The sending area of the cell starts right about here at this thick part of the axon called the axon hillock. The axon hillock is the area of the cell where the axon is really thick, where it's attached to the body. And this is where an electrical impulse is first generated. And then that electrical impulse is always sent away from the cell body. And it's going to end up in the terminal, causing this neurotransmitter to be released from these stored vesicles. Now, this axon may have branches to it. They're called axon collaterals. I don't talk about those all that much just because they mess up my drawing, but this can have a branch coming off of it sort of at a right angle like this, and that would be called an axon collateral. Some axons can have multiple collaterals to them. So the impulse that's generated at the hillock here can go, obviously, this direction as well as via these collaterals as well, be sent in slightly different directions. I'm just looking at my list here to make sure I went over all of what I think are the most important anatomical structures of this. Finally, this term, axolemma, refers to the plasma membrane of the axon. So I'm just going to write that out. This word, all that means is the plasma membrane of the axon area. And that's going to come into play when we discuss how this thing fires electrical impulse. So the axolemma in my drawing would just be the plasma membrane along here of the axon. So I need to draw a new neuron just because this one has gotten so complicated and busy to discuss. The next topic I'm going to discuss is what goes on when this thing makes raw material for a neurotransmitter in this area and then sends it this direction. So let's do that. I'm going to quickly draw another motor neuron. There's the nucleus. Just add some dendrites. Now, when this cell is at rest, meaning when it's not firing an electrical impulse, it's not sleeping, it's not inactive. As a matter of fact, it's got a high metabolic rate and it's very active. And what this cell's doing when it's not firing an electrical impulse is it's making the raw material in all of its organelles. So it's performing a lot of metabolism, it's making proteins, and it's shuttling that material in this direction toward the axon terminal. And that directional flow of material has a name. We're talking about something called transport of material along the axon. And this word, anterograde, what that word refers to is the flow of materials, in this case raw materials for neurotransmitter, in this direction from the cell body out here to the axon terminals. That's all the word anterograde, movement away from the cell body. Retrograde is just the flow of material in the opposite direction. That's the flow of material. I'll use a different color. Retrograde would be the flow of material from the terminal end back toward the cell body. And we can use, the neuron can use retrograde activity or flow of material when something down here needs to be recycled, for example, or reused to make more raw material, or just waste material from the assembly process can be sent back to the cell body where lysosomes, for example, could break that material down. So these two terms, anterograde and retrograde, refer to the flow of material, directional terms along the axon. Now, I need to talk about the myelin sheath. We mentioned that when we were talking about neural glial cells. Myelin is just an insulator, a fat-based insulator. So I'm going to write lipid-based insulation that's created by oligodendrocytes or oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. And it wraps around this insulation or these cells. If you were to look at this axon in cross section like this, these Schwann cells, what they do is they wrap around this axon like wrapping paper or like toilet paper on a roll, but they're fat-based cells, so they have a thickness to them. And they wrap in sections. So the myelin sheath looks like this wrapped around an axon. We've got an insulation, lipid-based material from either oligodendrocytes or Schwann cells that wrap around the axon and insulate it in this fashion. Notice that I left gaps in this. So this myelin sheath is along the axon, what your book calls nerve fibers. They use that sort of interchangeably with axons. Axons may, so I'm going to write wrap axons. Axons do not necessarily have to have myelin. Some do and some don't. But all dendrites are unmyelinated. So dendrites never have myelin. But axons may or may not have it, depending on the type of nerve, the type of neuron, where it's located, how fast transmission has to be. So here's what it does. The function of this stuff, I'm going to go back to green. It insulates, protects, and most importantly, not that the other two aren't important, speeds up transmission along that axon of the electrical impulse. I'll tell you how later. So just remember, in the peripheral nervous system, myelin is made from Schwann cells. In the central nervous system, myelin is made from oligodendrocytes. The significance of the differences of those two will come much later when I talk about damage repair should a neuron get damaged. So the thickness of the myelin depends on how many spirals or how many times it wraps around this axon. And believe it or not, the thickness of the myelin is going to dictate the transmission speed of the electrical transmission. So plasma protein channels are absent from Schwann cells. This is what makes it a very good insulator. So it's protein channels. These are very tightly knit around here and provides really good insulation and isolation. These gaps, finally, these gaps that I've left in the myelin sheath have a specific name. They are called nodes of Ranvier, just named after the French guy who discovered them. So that by definition, the nodes of Ranvier are gaps in the myelin sheath where I put little stars, okay? So here's a node, here's a node, here's a node. And what we're gonna find is when an impulse is generated at the axon hillock here, if this axon is myelinated, wrapped in insulation, the electrical impulse takes giant steps and it jumps from node to node. I'll talk about that process. That's what speeds up transmission. Before I leave today, I do need to start talking about membrane potential. I have to at least start it. I might not finish it. I'll start it here, and then I'll begin tomorrow's Zoom meeting with this same topic, because there's no way I'm going to be able to get through all of this in the next 5 or 10 minutes. So membrane potential refers to along the membrane of a cell, there's this, that's what the word membrane means, obviously. Potential just means there's a difference in the environment from this side of the membrane to this side. Something about this side of the membrane of the cell is making it different from this side. So that's what the strict definition of the word means. And that difference is basically based on the flow of ions through this membrane. And so the ion concentration is going to be different on one side of the membrane from the other. So membrane potential, those words, that's what it means. It just means there's a different ion concentration on this side than there is on this side. And that creates a difference. The word potential literally means the potential for energy or to do work. I always tell students, it's because it was handy for me when I was a student, think of a battery, a regular normal battery. You know that that battery has what we call a positive end and a negative end. And what we're doing inside, what has been done inside of this battery is we've separated ions and put a different concentration on this end than we have on this end. That's what we're doing to a cell membrane. We're altering the ion concentration on this side from this side. That gives the potential for energy or for it to do work. And what helps create this ion difference are channels, protein channels along this membrane. And there are basically 3 types of gated channels in the human body, on our cells. I'm going to list those 3. There are chemically gated channels. By gated, that means they open and closed based on chemicals that bind to them. There are also voltage gated channels that open and close based on a change in electricity or charge. And finally, there are mechanically gated channels. Just like the name says, that the protein gates open and close based on some sort of mechanical stimulation, like pressure or touch, for example. So those are the 3 types of gated channels. In this immediate discussion, we're going to be mostly concerned with these 2 types. And now I'm just going to set up the process or the topic of, this is the last topic, I promise, of resting membrane potential in a neuron, a nerve cell. So this just means membrane potential, which we just defined as being a separation of ions, creating the potential to do energy or work. And this is when the cell is at rest. So we're going to describe the membrane of the cell when it's at rest, all right? And it refers to a difference of ionic concentration across that membrane. So first we say the membrane, because there's a difference in ionic concentration, the membrane is what we say polarized. You know that polar means opposite or different. And it just means that there's different or opposite charge environments from one side to the other. So this only occurs along the edge of the membrane. So I'm going to draw a very simple nerve cell. Try not to complicate it. And this term, resting membrane potential, means that there's a difference in ion concentration on the outside along the edge of this cell than there is on the inside. So we have, during rest, a nerve cell has a high concentration of sodium ions all around the outside of the membrane. I'm not going to go all the way around. And it's got a very high concentration of potassium ions on the inside. That's due to a pump. It's called the sodium-potassium pump, and it's found on almost all of the cells in the human body. So I'm just going to draw that pump all along the membrane, and it uses ATP. It's an ATP-driven pump, so it uses energy. A lot of our energy in our body goes toward this pump on all of our cells. And what this little pump does, it's fueled by ATP, is it pumps sodium ions outside of the cell, and it keeps pumping potassium ions inside of the cell. See, ions always want to equalize. So if I put sodium here and potassium here, just by differences in concentration, by the way, these membranes are leaky. There's holes in the membrane. And sodium always wants to go down its concentration gradient in order to equalize through diffusion. Likewise with potassium going out. But we have this ATP-driven pump all along the membrane of our cells, specifically in this case, our nerve cells, that anytime sodium leaks through here, it grabs it and pumps it back out, so that we get a high concentration of sodium ions out and a high concentration of potassium ions in. Now, I know that sodium is positive and potassium is positive, but this is a much greater positive ion than this one. And so when we compare the environment of outside to inside, this side of the membrane is much more positive than the inside. And when we compare the two, so what we say is the net charge along the membrane at rest is positive outside and negative inside. The reason we say that is just because we're comparing the two sides to each other. So the net charge during rest of this cell is positive out, negative in. That's because there's a high concentration of sodium ions out here and a high concentration of potassium ions in here. And that pump is what creates it. And we say that this now has potential to do work or for energy. We're setting up an energized situation, just like that battery that you buy at the store stores energy by separating ions in it. And when you hook something up to it, guess what? It causes the flow of ions to move in the opposite direction. That gives off energy to your device. Same thing with this nerve cell. I am not going to expect you to get all hung up in the electricity in this chapter. So I want you to still read the chapter. Don't ignore the textbook. Students who read always do better than students who don't. But I am not going to expect you to get hung up on things like current and millivolts. Those are all just numbers and definitions. It doesn't matter to me what the value of negativity this negative is right here. Your book will try to say it's negative 70 millivolts and all this stuff. That's not critically important. What is important is the setup that I've highlighted here of what actually creates this resting membrane potential, okay? The last thing I'm going to say for the day is this. I'm going to define the word. I need a new whiteboard because I've already layered on 10. I'm going to define the word depolarization, okay? So on this last whiteboard, what does this word mean? Well, if along this nerve cell membrane, and I'm going to blow it up really big, literally what this word means is this. It means the change in membrane potential relative to resting membrane potential. So this just basically means a change in potential. That's its literal definition. In reality, here's what's going on. So if at rest, because we have tons of sodium out here, tons of potassium in here, and therefore a comparison, what we say the net charge is positive out, negative in. That's all the way around this cell. I'm just not going to draw it all the way around. What we've created here is a polarized situation at rest. So the resting membrane potential is polarized because of this, positive, negative. If we depolarize something, that means we make it less different. We make it less opposite. So picture this. Here's the globe, the Earth. There's the North Pole. Here's the South Pole. These are polar opposites. You know that. The act of, if you're standing on one pole and I'm standing on the other, we would be at polar opposite ends of the Earth. Now, if we both travel and migrate toward the equator, here, we are creating a less polar situation. We're getting closer and closer together, and we meet at the equator. That is the act of depolarizing because we're becoming less polar from each other, our location. Same thing applies to depolarizing a membrane. If we make this membrane that is positive out, negative in, less polarized, then we are depolarizing that membrane. So what's going to happen during depolarization is actually, just to cut to the chase, I'll start here tomorrow. Sodium ions out here, when they flow into the cell, that is an act of depolarizing the membrane because we're making the outside of the cell less positive and the inside of the cell less negative. So when sodium ions go into the cell, we depolarize. All right, so I'm gonna stop there. That's enough. It's crazy complicated information. If you have any questions, let me just shut off my recording.